Okay. Good afternoon, viewers at home. My name is Adeotunde Michael. I'm here this evening again with a repeat of the ongoing lecture on microbial genetics, with a very special focus again on microbial genetics. Okay, with a very special concern on what do we mean by gene regulation, as well as then we will spend time in talking about recombinant, that is, if there is time, recombinant DNA recombinant DNA technology. Okay, just like what we what I actually gave us on gene expression. I told you that why do we have some genes being synthesized all the time, whereas in the other gene we have very few genes that are being produced. There are some microorganisms that from time to time, every time, they will just be producing that. That is with respect to the function, how functional is that gene. And that's why I said any gene that is functional, that the microorganism or that organism need, that means that gene will be synthesized or expressed from time to time. So that's why some genes are more, or they are expressed more than the others. And I told you that in terms of gene regulation, or in terms of gene expression, it depends on whether the microorganism is a prokaryotic organism or is an eukaryotic organism. There is a different ballgame if the organism is prokaryotic, and a different, very um, little differences if that organism is an eukaryotic. So attention will now be shifted to a typical example of whatever we say today in terms of gene regulation, gene expression. It is either we are taking E. coli as a basic example, a basic representation of gene expression in E. coli. And if we are talking of eukaryotic, that is with reference to yeast, that is Saccharomyces cerevisiae. Okay, so um, that is that. Then I told you that an organism A, or organism, any organism, be it eukaryotic organism or a prokaryotic organism, we tend to express more genes that are vital. For example, gene that is helping an organism to fight against the host cell. That means that gene must be essential genes, must be an essential gene, and microorganism will tend to produce that the more. Genes that will make the organism to be able to ferment lactose. If the organism happen to be a lactose fermenter, that means the organism should make sure that it synthesizes genes or express genes that are very vital to that. So, and let me go back to what I said earlier on, and that's why I said when you look at DNA, which is very, very long like this, okay, this is gene expression, and don't forget DNA is double stranded. So at this level, the DNA will transcribe, is going to transcribe all the genetic information that you have in the DNA, is going to transcribe it into single stranded RNA. messenger RNA. And once that one is being transcribed to messenger RNA, then you now have a synthesis of polypeptides, or what we call proteins, or which could be in form of genes. So the genes that will be produced at this level, so the genes that will be produced at this level must be an essential genes that microorganism will actually need for the survival. So from mRNA to polypeptide is going to follow through a process that is called translation. We actually look at this the other time. So let's start with transcription. I just want to make mention of something, transcription. Okay, for transcription to proceed, it must proceed via four processes, which is initiation. Okay, after initiation, you have elongation. After elongation, you have termination. Okay, and you know exactly what happens at each of these stages. Don't forget that it is at the point of initiation that transcriptional factors will bind to promoter region. It's going to bind at promoter region, and at the point of starter box, when this won't bind, the process of transcription will be initiated. And by the time this one is initiated, elongation will now follow. And when elongation follows, that means that is when, at that point in time, that the double-stranded DNA will now be transcribed. Will now be transcribed with this. That's going to be unwinding the double-stranded RNA, and the enzyme that is actually involved in transcription, don't forget, it is called RNA polymerase. RNA polymerase. So that is the enzyme that is going to bind to the promoter region, and it is the same enzyme that is going to unwind the DNA double-stranded to be able to proceed via elongation step. Then it is at that level again, it is at the level of elongation that the T that you have in DNA will now be replaced by uracil. It is at the point of elongation that at least these two will occur: unwinding of the double-stranded of DNA and the replacement of thiamine with uracil through the help of RNA polymerase. And the last one, which is going to be the termination stage, is going to involve, that means the RNA will bind at terminator region. It's going to bind at terminator region, and once it has binded at terminator region, what is going to come out is still going to be single-stranded messenger RNA. It's going to be single-stranded messenger RNA, and it's going to be like three. We have a ribosomal RNA, messenger RNA, and we have a transfer RNA. All these three will now be produced at termination stage. Now, the fourth stage is called post-transcriptional modification. So, you know, immediately, for example, now if you visit a company that produces pure water, or let me say bottled water, so after the raw material has actually started, every other thing we proceed is going to move from one place to another, until it gets to the final stage, whereby the products will now be packaged. At the point of packaging it, that is exactly one of the reasons why there must be post-transcriptional modification. For example, maybe at a point of initiation, elongation, and termination, if some things were not properly done, or maybe one way or the other, there is a malfunctioning of any part of this, it is the function of this to be packaged modification. And just like I told you, there are two major events. There are two major events of transcriptional modification. The first one is called capping. The first one is called capping. That is, it's just like corking. You are adding capping. So, and capping will involve what is called a polyadenylation. Polyadenylation. That is addition of many adenine as the cap at the end of the messenger RNA. There will be addition of multiple molecules of adenine, which is a base. That is what is called, and that's the first post-transcriptional modification that microorganism will undergo, especially if you are talking of gene expression in prokaryotic organism. So after capping, the next one is going. Splicing is like cutting or removal. Okay. Now don't forget that whenever you have DNA here, okay, let me make a more illustration of what gene expression is like, okay, of what gene expression actually look like. Okay, let's say you have a long chain of a DNA, and on that DNA you now have all these segments. Let's say you have all these segments that are coding for special information. Okay, so wherever you see E, that is standing for exons. In that, these are coding sequence. These are coding sequences. And when you look at introns, these are non-coding regions of the lack or the operon of gene expression. So let's say you now have E at this point, E1. This is E2, E3. This E4. This E5. This E6. This E7. This is E8. This is E9. This is E10. Okay, don't forget that E is standing for coding regions. E1, E2. So the other one, these are non-coding. So it is, this is I1, I2, I3, sorry. Oh, this is I2. This will be I3. This is going to be I4, I5, I6, I7, I8, I9, I10, I11. So in this DNA, we have 10 exons regions, that is the coding regions, and we have 11 non-coding regions. So immediately this one has undergo process of transcription. You are still going to have it like from I1 to E10, and you are going to have it I1 to I11 is still going to be there. So immediately it is going to undergo initiation, elongation, and termination. So at the point of termination, it is now going to undergo post-transcriptional modification, which involve capping, which is a polyadenylation. So let's say you now have multiple addition of adenine, and you still have the messenger RNA like this that has E1 then down to E10. Then it also has from I1 down to I11. So the next thing after polyadenylation is called splicing, that is cutting, that is the removal of non-coding regions. That is what is going to happen, that all coding regions will be removed. So it's not going to leave us with what is going to have happen, and that's going to be removal of all I's, so that you now have E1, E2, E3, E4 up to E10. So, and that's the reason why the strand, the information of the strand that started the journey will be longer than the one that is going to end it after post transcriptional modification. But you have to note that removal of non coding regions from the expressed gene will only take place in eukaryotic organisms. Will only take place in eukaryotic gene expression. It's not going to take place in prokaryotic. So in prokaryotic, it's only capping that could also be observed. So that is at that point in time, that is of gene expression in prokaryotic organism. So don't forget that I told you under the process of transcription, the RNA polymerase is the enzyme that will catalyze the process of transcription. And process of transcription is going to involve four different stages: initiation, elongation, termination, and the last one is post-transcriptional modification, which is going to involve poly, which is capping, polyadenylation, which is going to be addition of many adenine molecule, and splicing, which is going to involve splicing, which is the removal of coding and non-coding regions. Okay. Yeah. Now the next stage is translation. The next stage is translation. The next stage is translation. The next stage is translation, and that is translation is synthesis, the process by which proteins are being synthesized. That is the stage that is going to happen. Which is going to be synthesis of protein. So that one will also take place in three different conditions as well. So the first one is also initiation. The second one is also elongation, and the third one is termination as well. Each of these process, don't forget that at the end of transcription, we now have three different RNAs, and this is ribosomal RNA, which is like the cooking pots. So this is ribosomal RNA, which is actually divided into small subunits and large subunits. The large one in bacteria is 70S, whereas the small unit is called 30S. So it is at this 30X that you have with messenger RNA. On the messenger RNA, you have triplet codons like this, that each of these is going to code for different amino acid at the end of the day. Then here, so this is a messenger RNA, is the ribosomal RNA. So inside the ribosomal RNA is where you have the transfer RNA, which has the structure like this, which has this structure. Okay. So on the point of message of the transfer RNA, that's where we have the anticodon region. The codon region RNA, whereas anticodon is found on the transfer RNA, whereas it is the ribosomal RNA that is the factory that actually synthesizes protein. That is the factory that house. So, and transfer protein synthesis can only, this one can only contain two transfer RNA at the same time. So, and the one that is going to initiate it, the codon, is methionine, which is always AUG. Methionine. So AUG will be here. Okay. Now let's say now have this AUG, CCU, CAU, GCC, CAC, and if this one is amino acid 1, 2, until you have amino acids, which could be UAA, UAG, or UGA. That is this, any of these three can act as stop codon. The start codon is AUG, which is methionine. Now, this is going to be present on the messenger RNA. It's going to line up on the messenger RNA, whereas the anticodon, this one is going to be complementary to A to U, U to A, G to C. So this will be found on the anticodal region. So it is at this point that the anticodal region of transfer RNA now bind to AUG. So that is initiation. That means the process of protein synthesis has been initiated. So that means once this one has produced methionine, so this one is going to move out, is going to be replaced with the next one, which is CCU. Two at the same time, this one will shift to this place, another one will replace. Immediately that one has finished its work, this one will move in, another one will replace. So it's going to be like that until it has encountered any of the stop codon, and that is called termination. That's the point at which protein synthesis will be terminated. At that point, the protein synthesis is going to be terminated. Now, the fourth process, just like transcription as well, this one also has the fourth one, which is called post-translational modification. Post-translational modification. Okay? What happens at this point is either the polypeptide or the amino acid will become phosphorylated, that is through the process called phosphorylation or glycosylation. So phosphorylation is addition of a phosphate group. Whereas glycosylation is addition of what? Glucose. Addition of sugar or carbohydrates to the protein. And don't forget that depending on the destination of that protein, if that protein is meant for any part of the cell membrane, or if it's going to stay at the eyesight, that is going to serve the function of signal transduction, that is cell to cell communications. It is glycosylation that will be favored. But if it is going to be used, for example, in glycolysis or cellular respiration, it's going to favor more of a phosphorylation, because that is going to be involved in energy generation. So that is the last thing that will take place under after post-translational modification in form of phosphorylation and glycosylation is called the cleavage, until the protein, the product, which is now polypeptide, or is now right to be called protein now, until it has undergone that is called cleavage. Excuse me. Until it has undergo that process, that means the protein cannot become functional. That protein cannot become functional until it has actually undergo the process of cleavage, that is post-translational modification. Okay, now, so that will now terminate the process of gene expression. It's not going to terminate the process of gene expression. That means that gene, that functional gene, if it is going to be responsible for whatever, if it is an essential gene, it is going to undergo the process of transcription, then translation. And at the point of each point or each process, that means what is going to happen is going to be that. Okay. Now, that is now the end of gene expression. Gene expression. So let's now look at our main work today, which is going to be the how is gene being regulated. How is gene being regulated? Let me go just cite an example. Let me use very simple analogy.